Hello students, today in this module I am going to discuss about respiration in plants and we will be focusing on the topic electron transport system. So let's begin with. So we are studying the respiration in plants and we know that during the respiration there is a combustion of glucose. The glucose is converted into carbon dioxide, water and energy. These are the products. The energy is given out in the form of the heat and the end products are the carbon dioxide and water. Okay, and these products are obtained by the complete oxidation of the glucose. Okay, this is the general reaction or the reaction for the respiration. Now, the respiration can be occurring as aerobic respiration or anaerobic respiration. Now, the difference between the two is that the aerobic respiration yields more energy and it takes place in the presence of the oxygen, and the respiratory substrate here is completely oxidized. Okay, the respiratory substrate, which is the glucose, it has been completely oxidized and this takes place in the presence of the oxygen, and here more energy is released. If I talk about the anaerobic respiration, here the substrate is partially oxidized and it occurs in the absence of oxygen. If I compare the anaerobic respiration with the aerobic one, the anaerobic yield less energy as compared to the aerobic here because here the oxidation is done partially. Now let us look at the mechanism of the respiration. Now, whether it is aerobic respiration or it is anaerobic respiration, the both start with the first cycle, which is glycolysis. I have already discussed glycolysis in the class, okay, and we know that in the aerobic respiration, first there is glycolysis, okay, and after the glycolysis there is a cycle which is known as citric acid cycle, and after the citric acid cycle there is electron transport system, and these two cycles, which is citric acid cycle and the electron transport, they both occur in the mitochondria, which is known as the powerhouse of the cell. I am repeating that the aerobic respiration involves the very first cycle, that is glycolysis. It takes place in the cytoplasm and then it is followed by the citric acid cycle and the electron transport. The citric acid cycle and the electron transport takes place in the mitochondria. So these are the cycles which are involved in the aerobic respiration. If I talk about the anaerobic respiration, in which the substrate is partially oxidized, glycolysis is there, okay, in the cytoplasm, and then it will be followed by the formation of either the organic acid like lactic acid or ethyl alcohol, okay, and these two products are formed in the cytoplasm and they are formed by the process of fermentation, okay. So in the anaerobic respiration, oxygen is absent. Glycolysis is the first step for the partial oxidation of the substrate, and after the glycolysis in the cytoplasm there is formation of either alcohol or organic acid, okay, and the process of forming the alcohol is known as the fermentation. Now let us discuss the first cycle, that is glycolysis. This topic we have already done in the class, so I am just going to give a recap of that. Okay, in glycolysis we are having the first substrate, we are having our substrate, that is glucose. The glucose is a 6 carbon compound, okay, and it has been converted into glucose 6-phosphate, then fructose 6-phosphate, then fructose 1,6-bisphosphate, and then triose phosphate or triose phosphate dihydroxyacetone phosphate. Okay, here we can see here was the 6 carbon compound, now here it is, these are the 3 carbon compounds, okay, and they are able to convert to each other. Okay, now one more thing, in the complete glycolysis there are many enzymes which are involved. Each step is catalyzed by a particular enzyme, okay, and in these steps we are at the end getting the pyruvic acid. Okay, so glucose, when it is converted into triose phosphate or dihydroxyacetone phosphate, in these steps 2 ATP molecules are used. Okay, and ATP molecules are used to convert the glucose into these particular products. Okay, and in this part of the cycle, what happen is one NADH molecule is produced and 2 ATP are produced. Water is also released. Okay, so I can say here 2 ATPs are used and here 2 ATPs are produced, and here NADH molecule is been released. Okay, now one thing here to be noted, that one glucose molecule is forming the 2 molecules of the pyruvic acid. One glucose molecule is forming 2 molecules of the pyruvic acid. So as a whole, during the glycolysis as a whole, there is production of a net gain of 2 ATP molecules from one glucose. As one glucose molecule is making 2 pyruvic acid, okay na, 2 pyruvic acids are made. That means, what does it mean is that here 2 ATPs are used in these steps only. Okay, and here if I calculate for the 2 pyruvic acids, means there are 4 ATP molecules which are produced in the other half of the cycle. Okay, if I talk about the 2 pyruvic acid, in the formation of the 2 pyruvic acids, a total of 4 ATP molecules are formed, and on the upper stages of the cycle 2 ATP molecules are used. So as a whole, if I minus the both, I will get 2 net gain of the 2 ATPs molecule directly. Okay, so in the glycolysis we are having the net gain of 2 ATP molecules. Okay, and we are also getting 2 NADH molecules. Okay, so at the end I can say, from the complete glycolysis, the glucose, the 6 carbon compound glucose get converted into 2 pyruvic acid molecules, and in this complete cycle we have the net gain of 2 ATP molecules and 2 NADH molecules. Okay, now this cycle of glycolysis is common for both aerobic and anaerobic type of respiration, and it takes place in the cytoplasm. Now if I talk about the anaerobic respiration, after the glycolysis in the cytoplasm we are having the pyruvic acid. Now this pyruvic acid, now in the absence of the oxygen, it can form either the organic acids, that is like lactic acid, or it can make ethanol and carbon dioxide by the process of fermentation. Okay, so these are the products that we will get in case of the anaerobic respiration. Let us talk about the aerobic respiration. Now as I told you, in the aerobic respiration, after glycolysis, the pyruvate is transported from the cytoplasm into the mitochondria, which is the powerhouse of the cell, which is regarded as the powerhouse of the cell, and the next steps of the cycle take place in the mitochondria. Okay, and the product, that is pyruvate, is been converted into acetyl CoA first. Okay, the pyruvic acid, the pyruvate, which is a 3 carbon compound, it is converted into acetyl CoA, and during this reaction there is formation of NADH and carbon dioxide gas is released. So as we know, from the one molecule of the glucose we have got 2 molecules of the pyruvate. Okay, and 1 molecule of the pyruvate is giving you 1 molecule of the NADH. So I can say, during the oxidation and during the reaction of the pyruvate to form the acetyl CoA, the net gain of the NADH molecules are 2. So we are getting 2 molecules of the NADH as a whole from 2 molecules of the pyruvate. Now after this step, after the formation of the acetyl CoA, which is a 2 carbon compound, okay, The next is that the acetyl CoA enter into a cycle, and this cycle is known as the citric acid cycle. We also call it as the Krebs cycle. During this cycle, what happens is the acetyl CoA combine with the oxaloacetic acid, and it make the citric acid. This citric acid then is converted into many intermediates by many reactions. Okay, many steps are there total, and during this complete cycle, there is net gain of 6 NADH molecules. There is net gain of the 6 NADH molecules, 2 FADH2 molecules, and 2 molecules of the GTP. Okay, so these are the end net gain of the molecules from the citric acid cycle. I am telling about the 2 molecules of the pyruvate. I am not considering a single molecule of the pyruvate. Okay, if I talk about the single molecule, then you can see that in the citric acid cycle there is formation of 1, 2 and 3. 3 NADH molecules are produced from a single pyruvic acid. So if I double it from the 2, it will be made 6 NADH molecules. Similarly, we can see in this cycle there is formation of 1 molecule of FADH, and so the 2 molecules of the pyruvic acid will be making 2 molecules of the FADH. There is net gain of GTP over here, 1 molecule, and as I am talking about 2 pyruvates, so 2 GTP molecules will be produced. This citric acid cycle takes place in the matrix of the mitochondria. Now after the citric acid cycle, the next thing is the electron transport system. Okay, now these are the steps in the respiratory process, and they are used to release and utilize the energy which is stored in the NADH and FADH2. We already have the net gain of few ATP molecules already, like 2 molecules we have got directly from the glycolysis, and we have got 2 molecules of the ATP during the citric acid cycle. Rest of the molecules are in the form of the NADH and FADH. Okay, and we need to utilize them, and we need to release the energy out of them. Let I tell one more thing. 1 molecule of NADH will give you 3 molecules of the ATP. After the complete utilization of the NADH molecule, from 1 molecule we will get 3 molecules of the ATP. Okay, now let us see how these 2 molecules, that is NADH and FADH2, are used, and how the energy is released from them, and the energy is released with the help of the electron transport system. Now this is done when they are oxidized through the electron transport system, and the electrons are passed to the oxygen, and it results in the formation of the water. So whatever electrons are released from NADH and FADH, okay, they all are transferred to the oxygen, because it is the oxygen receiver over here. Okay, it is the oxygen carrier, and it, with the help of those electrons, it utilize them, and it utilize the protons, and it make the water. Now the metabolic pathway through which the electrons are passed from one carrier to the another carrier, that is known as the electron transport system. Okay, and it is present in the inner mitochondrial membrane. If I talk about the structure of the mitochondria, you must be knowing that the mitochondria is a double membraned organelle, and it is having the outer membrane and the inner membrane, and in between two membranes there is present the intermembrane space. Okay, and inside the inner membrane there is present matrix. So this is the complete scheme which is shown over here. This is the matrix, the inner matrix, and this is the inner mitochondrial membrane. This is intermembrane space, and after the intermembrane space, here will be the outer membrane. And this electron transport system take place in the inner mitochondrial membrane. whereas the cycles which we have already discussed, that is the citric acid cycle, okay, and the conversion of the pyruvate into acetyl CoA, that all happens in the matrix, okay. Now let us discuss about the electron transport system. The electron transport system consists of certain complexes. There are a total of 5 complexes. The first complex is known as the NADH dehydrogenase complex 1. The complex 1 is NADH dehydrogenase. Complex 2 is known as succinate dehydrogenase. Complex 3 is known as the cytochrome BC1, and complex 4 is known as the cytochrome C. Complex 4 refers to cytochrome C oxidase. So we are having these 4 complexes in the electron transport system. The fifth complex, which I am telling you, it is actually the ATP synthetes. It is regarded as the fifth complex, or we can also call it as the ATP synthetes. Now let us discuss how the electron transport system works. Now NADH, which was produced during the glycolysis and during the citric acid cycle, they are oxidized by the NADH dehydrogenase, that is the complex 1, and the electrons that are produced during this, they are transferred to the ubiquinone. Okay, these electrons which are released during this during this reaction, okay, during this reaction, they are transferred to the ubiquinone, and they are transferred via FMN. The ubiquinone, which is the intermediate carrier, it also receives some set of electrons, equivalent amount of the electrons, via FADH, which is generated during the oxidation of the succinate by the succinate dehydrogenase. Okay, so this ubiquinone, these ubiquinone, okay, they are receiving the electrons, similar number of the electrons from complex 1 as well as from the complex 2, that is succinate dehydrogenase. Okay, and the reduced ubiquinone, after getting the electrons, the reduced ubiquinone is known as ubiquinol, and it then oxidized by transferring the electrons to the complex 3, which is known as the cytochrome BC1. So this ubiquinone is transferring the electrons to the complex 3, and here also it is transferring the electrons to the complex 3. So whatever electrons are received by ubiquinone from complex 1 and complex 2, they all electrons are transferred to the complex 3, that is cytochrome BC1. Okay, now after this, from the complex 3, the electrons are transferred to cytochrome C. All the electrons are transferred to the cytochrome C. Okay, and this cytochrome C act as a mobile carrier. Okay, it connect the complex 3 with complex 4. Okay, so complex 3 and complex 4 are connected to each other. Okay, and there is a connection between them via cytochrome C. Now this cytochrome C have got all the electrons coming from the complex 3. Now it will transfer the electrons to the complex 4. The complex 4 is containing cytochrome A and cytochrome A3 as well as 2 copper carry centers. It is having 2 copper centers, cytochrome A3 and cytochrome A. Okay, when the electrons are shunted over the carrier via 1 to 4 in the electron transport chain, they are coupled to ATP synthetase, that is the 4th complex for the formation of the ATP from ADP and PI. So the transfer as well as the formation of the ATP is coupled over here. and whatever protons are, whatever electrons, okay, they are produced, they all are passed to the oxygen, okay, the electrons which are been transferred from these complexes, from complex 1 to ubiquinone, then complex 3, then from complex 3 to cytochrome C, and from cytochrome C to complex 4, they all are transferred to the oxygen, which is the electron, which is the terminal acceptor of the electron, okay, and it is also receiving the protons which are coming from here. These protons are released, okay, and the oxygen, on receiving the electrons, it get reduced to water along with the hydrogen atom, okay, it react and it make the water molecule. So I can say that the oxygen is the terminal acceptor, and the oxygen function as a terminal acceptor of the electrons, and it is reduced to water along with the hydrogen atoms. Now, as I told you that the fifth complex, that is our ATP synthetes, it is also coupled with the transfer of the electrons from one to another complexes, and formation of the water is there from the oxygen. So it is also working simultaneously. Okay. Now we can also see from the diagram that there is increase in the proton concentration in the inner membrane space as compared to the outer, as compared to the matrix. So here the concentration of the protons is more if I compare it with the matrix, okay, and these protons are then moved from inside, from the inner membrane space into the matrix, okay, and there is a formation of the electrochemical gradient over here. You can see here is more positive charge, here is the negative charge. Reason being, inside there is, in the inner membrane space, there are more number of the protons which are released during the transfer, okay, and on the matrix there is less number of the protons, so an electrochemical gradient is formed. Now let us discuss about the ATP synthetes. The ATP synthetes consist of two major components. The first is the F1 and the another one is the F0. The F1 is called the headpiece, and it is the peripheral membrane protein complex, and it contains the site of the ATP formation from ADP and the inorganic phosphate. So the ATP synthetase, that is our complex 5, it is consisting of the two components F1 and F0. The F1 is known as the headpiece, and it is at the peripheral membrane protein complex, and it is the site where ATP is produced from ADP and inorganic phosphate. If I talk about F0, it is an integral membrane protein complex, and it is a channel through which protons cross the inner membrane. The passage of the protons through the channel is coupled to the catalytic site of the F1 component for the formation of the ATP. And for each ATP molecule which is produced, 4 H positive, that is 4 protons, are passed through F1, F0 from the intermembrane space to the matrix down the electrochemical proton gradient. So from this diagram, it is already very clear. This is the outside, okay, this is the intermitochondrial membrane, and this is the matrix. I told you that ATP synthetase is having two parts. One is the F0 and F1 and the F0. The F0 part is present in the intermitochondrial membrane, whereas the F1, which is the head region, it is present in the matrix, and it is the point where ATP is formed by utilizing ADP and the inorganic phosphate. And the F0 work for the transfer of the protons from the outside to the matrix, and for each ATP molecule 4 H positive, that is 4 protons, are transferred. So after this, we know about now we have got the ATPs out of all the glucose, and glucose is completely oxidized at the end of this ATP synthesis working, which we also call it as oxidative phosphorylation. Okay, so after the synthesis of the ATP, okay, we have done with the complete oxidation of the glucose, and this is the net gain of the ATP from the one molecule of the glucose. So there is a net gain of 38 molecules of the ATP as a whole during the aerobic respiration, okay, of the one molecule of the glucose. So from one molecule of the glucose we will get 38 molecules of the ATP. Okay, and the glycolysis, as I told you, from glycolysis we are getting 2 ATP molecules directly, and we were getting 2 NADH molecules which will enter the electron transport chain, and we will get 6 ATP molecules out of them. From 1 NADH we will get 3 ATP. And from the second cycle that we have discussed, the pyruvic acid to acetyl CoA, from this reaction 6 ATP molecules will be formed. From the citric acid cycle we will get 18 ATPs from 6 molecules of the NADH, 4 ATPs by utilizing the 2 molecules of FADH, and 2 molecules of the ATP directly. So as a total we have got 38 ATP molecules. Now, as we know that in the eukaryotic cells, 2 molecules of ATP are spent for transporting the 2 molecules of the NADH that is formed during the glycolysis in the cytoplasm to the mitochondria. So the net gain is 36 molecules. I am explaining this again, that we know that the glycolysis is occurring in the cytoplasm, and after the formation of the 2 molecules of the pyruvic acid, some energy was required to transfer these pyruvic acid molecules into the mitochondria, and during this transfer 2 ATP molecules were used. So so we can minus 2 ATP molecules from this 38 net total. Okay, so we have got 36. So I can say that the net gain of ATP from the complete oxidation of the glucose during the respiration is 36 molecules of the ATP. I hope, students, you have understood about the respiration in plants and how the mechanism takes place of the aerobic and the anaerobic respiration. Thank you, all of you.